Hey all, so here's the week 7 lab. We've got 2 of them, genetic inheritance. Hopefully you started all this stuff earlier, last week at least. Please, please, please say you did. And then we're going to have the inheritance lab formatted and the rubric. You won't see the faculty materials. Please share any tables in case someone's stuff didn't germinate correctly, because, you know, life may find a way, but sometimes it won't be in our 7-week class. Let me pause this and open up the rubric first. Okay, here's the rubric for the lab on genetic inheritance. So we got 3 exercises at 1 1/4 point each, 3/4 a point for the tables, and then you will have your grammar at 1/2 a point. So it's pretty standard, lets you know there's 3. Let's go over and walk through the lab. I already have it started on. Right, just in case I didn't record that part, we got 3 exercises, 1 1/4 each, 3/4 for the tables. Let's go over now to the lab walkthrough. Okay, so we got the title page, and then it's going to have genetic inheritance. Okay, this is the table where you're going to say what the dudes look like. It's using big G, little g. If it doesn't tell you anything, you can use whatever you want to. And then you have how many you found that were green and white. I'd use big G for green. White, little g, just easier. Let's see, anything else? No. Then we fill in our expected, our Punnett square, effectively. Okay, so up here you'll put whatever you have, and down here you'll put on the other side. So the F1, what will the gametes be? F2, what will their gametes be, right? And then you can say, like, yeah, here's, like, you know, big G over here, little g, little g. There you go. Or PS, whatever makes you happy, right? So you'll have the F1's genotype, the other F1's genotype, and then you'll put it in here. So if it was big P, big S, little p, big S, whatever, and then you'll say what they would combine to create. Okay? Okay. And we're doing PSs, so that's what it's going to be. Then down here, we are going to have, okay, so you'll say, what's the color? So basically, it's going to be either purple or yellow and smooth or wrinkled, right? And it'll be some combination thereof. It has an example of yellow and wrinkled being the recessive, so that's what we want our recessive to be. And then you'll put in what it looks like, descriptive-wise, what the genotype is, because with phenotype, genotype, then what we would expect to look at 2 traits that are not linked, what the percentages should be, and then what the number we actually saw and what that equals as a percentage. Hopefully, you will have, well, you have 100. So percentage, and if you see 34, that would be 34%, or the frequency would be 34 over 100, or 0.34. If that was it, probably be some crazy thing. Then D to 2. It's going to be the same thing. You talk about your phenotypes. Then you're going to do how many you saw from this, from the number, the ratio, the expected ratio. Over here, the expected number calculated. So if that was the ratio, what should you have seen? If it was like 50 over 100, you should see 50, right? And then you're going to take the observed number minus the expected number, figure that out, square it, so times it by itself. So if this was 10 minus 6, you would have 4. 4 squared, or 4 times 4 is 16, divided by the expected number, which I gave you was 6 in this thing, so it'd be 16 divided by 6. I'll give you some decimal. And then down here you'll see the sum of all of them, you had 100. And then over here what your chi-squared value is. And it shows you what that is over here, right? Same thing. So you'll just add these all up and get something. I think. Let me check. I'm adding. That sounds right. Where's the third one? It's got 3. The sum, oh no, it's the sum of the observed number minus the expected number squared minus that. Okay, so it's basically that. But we take all of those added together. So you take all of these add them together, and then divide by the expected number. Okay, where is it? So you take all that before you divided it, add all that sucker together with the squares. So it'd be like the 10 minus 6 is 4 squared the 16, and you'd add that up for all the different ones, and then you divide, after you added all of those together, you would divide them by the expected number. Alright, it's chi-squared. We bring it down. We have a value of 3 degrees freedom. So then what's the probability? And is it a good fit or not? You just circle one. You can just delete the other one. Yeah, just delete the other one. Sorry. Okay, so then let's get into, it will walk you how to do all that stuff in the lab manual pretty clearly, if I remember correctly. So then we're going to say, what's the expected phenotypic ratio of green to white? You'll let me know. And then what's our number? What's our seedlings equal? Okay, then we do, if you had 320, then how many would be green? How many would be white? Right, that's interesting. Discussion, free sports, it's a Punnett square. I love a Punnett square. I need to remember to post my Punnett square video in week 7. I will put a note. I have a good one. And then if the results are not similar, why are they different? Tobacco, will a mono hybrid cross of the corn be the same as the tobacco? So basically, does it matter what you're breeding? Will the same principles apply? You're going to look at your data from everybody else too, see what happened. Thank goodness, right? Construct a hypothesis. Record your hypothesis there. So, what are the 2 hypotheses that you made for these crosses? So parent times parent, what did you say about the F1s? Here's the F1 times F1. What did you say about F2 cross with F, or F1 cross with F1? What did you say about the F2s? What do you expect to see? Based on what you know, what's your genotype going to be? So this is back to your P's and your S's, right? If one's completely, if it's all dominant, if it's all recessive, does it make a difference if you cross these crazy ones? From the phenotype of the kernels and the P generation, what would be predicted of the F1 plant? Okay, and then we are up to, given the 2N equation, predict how many different genetic outcomes would be possible. So 2 to the nth, and they're saying that in a dihybrid, so how many suckers are you crossing? And then here we go. Has 536. What do you expect the F1 to look like? So what's your ratio, and use that to figure it out. Then how well does stuff predict? Let's look at C. Okay, it says progeny phenotypic frequency holds true universally. Explain, based on the experimental outcome, what is it? So what should it have been, and were you pretty close, kind of thing. Why might there be differences? So more or less like, when do you think the ratio will hold the most true? Does it depend on like maybe sample size? Application, so how would you apply it? How might the information be applied medically? Here's more on our chi-square. So you walk through, how can you test the fitness for other stuff? Give an example. Expected genotypic process, give an example, no problem. The total population is 6. Dear God, that's kind of tiny, huh? Where'd you go? We are at letter E. Okay, so you would take, what's your frequency? What was that percentage? That decimal, and multiply together. If the progeny population was 20, same concept. You still have this, you should have that same ratio. 1,000. Excluding factors such as sex-linked, incomplete dominance, etc. are used. Will the above cross result, does it matter if it's a dog or a tulip? Oh, how will? Do we not exclude those? We excluded them. Yes, so then how will that work? Does it matter? And then if you do bring in that stuff, so obviously this is going to be yes or no, and this will be the opposite. I'm guessing since you're excluding stuff, it's going to say it doesn't matter. That you bring that stuff in, it does. Why? Okay, hopefully this helped. It's going to be a lot of fun with Punnett squares. Hopefully everybody's plants grow. We will find out, huh? I hope it helped.